Hello, welcome to Biology, the science study of life. Motility. About half of all prokaryotes are capable of directional movement. Some species can move at speeds exceeding 50 diagrams each second, up to 50 times their body length per second. Of the various structures that enable prokaryotes to move, the most common are flagella, which may be scattered over the entire cell surface or concentrated at one or both ends of the cell. The flagella of prokaryotes differ from those of eukaryotes in both structure and mechanism of propulsion. Notice this figure. Prokaryotic flagella are 1/10 the width of eukaryotic flagella and are not covered by an extension of the plasma membrane. Notice that differences. In a relatively uniform environment, flagellated prokaryotes may move randomly in a heterogeneous environment. However, many prokaryotes exhibit taxes, movement toward or away from stimulus, from the Greek taxes, or to arrange. For example, prokaryotes that exhibit chemotaxis respond to chemicals by changing their movement pattern. They may move toward nutrients or oxygen positive chemotaxis, or away from toxic substance negative chemotaxis. In 23 scientists Princeton University and the Institute Curie in Paris demonstrated that solitary E. coli cells exhibit positive chemotaxis toward other members of their species, enabling the formation of colonies. Internal and genomic organization. The cells of prokaryotes are similar than those of eukaryotes in both their internal structure and their genomic organization. Prokaryotic cells lack the complex compartmentalization found in eukaryotic cells. However, some prokaryotic cells do have specialized membranes that perform metabolic functions. These membranes are usually enfoldings in the plasma membrane. The genome of a prokaryote is structurally very different from a eukaryotic genome and has on average only about 1,000th as much DNA in the majority of prokaryotes. Most of the genome consists of a ring of DNA that has relatively few proteins associated with it. This ring of genetic material is usually called the prokaryotic chromosome. Unlike eukaryotic chromosomes, which are contained within the nucleus, the prokaryotic chromosome is located in a nucleoid region, a part of the cytoplasm that appears lighter than the surrounding cytoplasm in electron micrographs. In addition to a single chromosome, a typical prokaryotic cell may also have much smaller rings of DNA called plasmids, most consisting of only a few genes. These plasmids genes provide resistance to antibiotics, direct the metabolism of rarely encountered nutrients, or have other such contingency functions. In most environments, the prokaryotic cell can survive without its plasmids, since all essential functions are encoded by the chromosome. But in certain circumstances, such as when antibiotics are used to treat an infection, the presence of survival of a plasmid can significantly increase a prokaryotic's chance of survival. Plasmids replicate independently of the main chromosome, and many can be readily transferred between partners when prokaryotes conjugate. And replication, transcription, and translation are fundamentally similar in prokaryotes and eukaryotes, although there are some differences. For example, prokaryotic ribosomes are slightly smaller than eukaryotic ribosomes and differ in their protein and RNA content. These differences are great enough that certain antibiotics, such as erythromycin, and tetracycline bind to ribosomes and block protein synthesis in prokaryotes but not in eukaryotes. As a result, we can use these antibiotics to kill bacteria without harming ourselves. Reproduction and adaptation. Prokaryotes are highly successful in part because of their potential to reproduce quickly in a favorable environment, dividing by binary fission. A single prokaryotic cell becomes two cells, which can become 4, 8, 16, and so on. While master prokaryotes can divide every 1 to 3 hours, some species can produce a new generation in only 20 minutes under optimal conditions. In reproduction continued unchecked at this rate, a single prokaryote could give rise to a colony outwinning Earth in only 3 days. In reality, of course, prokaryotic reproduction is limited as the cells eventually exhaust their nutrient supply, poison themselves with metabolic wastes, or are consumed by other organisms. Prokaryotes in nature also face competition from other microorganisms, many of which produce antibiotic chemicals that slow prokaryotic reproduction. The ability of some prokaryotes to withstand harsh conditions also contributes to their success. Certain bacteria, for example, can form resistant cells called endospores when an essential nutrient is lacking in the environment. The original cell produces a copy of its chromosome and surroundings it with a tough wall, forming the endospore. Water is removed from the endospore and metabolism inside it comes to a halt. The rest of the original cell then disintegrates, leaving the endospore behind. Most endospores are so durable that they can survive in boiling water. To kill endospores, microbiologists must heat their lab equipment with steam at 121 Celsius degree under high pressure in less hostile environments. And the spores can remain dormant with viable for centuries, able to rehydrate and resume metabolism when they receive, because that the environment has become more benign. Prokaryotes can adapt quickly to changes in their environment through evolution by natural selection because of prokaryotes' rapid reproduction. Mutations that confer greater fitness and swiftly become more common in a population. For this reason, prokaryotes are important model organisms for the scientists who study evolution in the laboratory. For example, Richard Linsky and his team have maintained colonies of E. coli through more than 20,000 generations since 1988. The researchers regularly freeze samples of the colonies and later thaw them to compare their characteristics with those of later generations. Such comparisons have revealed that the colonies today can grow 60% faster than the 1988 colonies under the same environment conditions. Linsky's team is exploring the genetic changes underlying the colonies' evolutionary adaptations to their environment. In 2023, they reported that 2 colonies showed parallel changes in expression for the same 59 genes compared to the original colonies. The rapid reproduction of E. coli enabled the scientists to document this example of adaptive evolution. Horizontal gene transfer also facilitates a rapid evolution in prokaryotes. For example, conjugation can permit the exchange of a plasmid containing a few genes or even large groups of genes. Once the transferred genes are incorporated into a prokaryote's genome, they are subject to natural selection during subsequent rounds of binary fission. Horizontal gene transfer is a major force in the long-term evolution of pathogenic bacteria, a topic explored later in this chapter.